Okay, so if you remember from our last steps, as a quick recap, we know that so far we have a little bit of we have 2 from glycolysis, 2 from the Krebs cycle, and that's a grand total of 4. We need 36 to 38 to keep us alive, so that's clearly not enough. If you're a bacteria, that's just enough to keep you alive, and if food goes away, you're gone too. But we have all these other enzymes, and those are those coenzymes. Those are going to be like those NAD that got turned into NADH in glycolysis. They're going to be the NAD that got turned into NADH into the prep cycle. In the Krebs cycle, we went ahead and made a total of 6 NADHs and 2 FADHs. And those guys, all in total, are going to be 10 NADHs, again, 2 plus 2 plus 6, 10 NADHs, plus 2 FAD. And we made that in all of these other steps. So we can use that to get more energy. And how that works is this. We're going to go ahead and take those coenzymes, the NAD and the FAD, and they're going to give us electrons and hydrogen ions. And that's going to help us make ATP. It's aerobic, which means it needs oxygen to go. The only one that hasn't needed oxygen to go was glycolysis. It occurs in these special areas in the mitochondria. This is called oxidative phosphorylation because we're going to need oxygen for this process, and we're going to use a different technique. Chemoosmosis occurs, which just means that a chemical sort of is going to go from one side of the gradient to another, and it's going to make some energy for us. And oxygen is the final electron acceptor. So let's talk about it. So this looks like a lot, but really it's not that bad. And if you remember my example from class, we talked about this being like the Hoover Dam. And the Hoover Dam is basically an area where you have a lot of water on one side and very little water on the other side. They have these turbines where the water could come down really quickly and turn some turbines. And when the turbines turn, because of that speed of the water coming down sort of like a ramp, it basically makes electricity, energy. So now if we look at this, let's keep equating it back to the Hoover Dam, and it'll make sense. So here we have NADHs. Remember we made those in a couple of steps. We made them in glycolysis, we made them in Krebs, we made them in the prep stage. Well, all of those guys, I kept saying that they're like they're going to go somewhere. This is where they go. They go to the sort of the inner parts of the mitochondria, and what they're going to do is they're going to drop off these hydrogens. And these hydrogens are going to go this direction. Also, they're going to drop off these electrons, and the electrons are going to go this direction. Let's ignore the electrons for right now. Let's just stick to the hydrogen ions. So the hydrogen ions for all those 10 NADHs come here, and the 2 FADHs come here per glucose molecule. And here the same thing is going to happen. These hydrogen ions are going to go up. The electrons are going to sort of continue this way for these guys because they're going to donate hydrogens and electrons as well. So now what happens is we have a high concentration of these hydrogen ions at the top, and we have a low concentration of hydrogens at the bottom. So what happens is all these hydrogen ions, they're going to eventually come to this. And this is an enzyme called ATP Synthase. So we know that anything that ends with an ASE is an enzyme, and ATP is energy, and syntha means making. So what happens is this is that turbine that's like in the Hoover Dam, that when water comes down it spins really quickly and creates some energy to do some work. Well, that's what's happening here. Hydrogen ions are coming down really fast, and it's turning this enzyme, and this enzyme basically takes that lower energy form, ADP, plus a phosphate, and sticks them together to become ATP, the triphosphate. So this is the high energy form, this is the low energy form, and when you slap them together, this becomes ATP. This could go somewhere in the cell to do some work, and when it does some work, it returns back in this version, the ADP plus a phosphate, and they just get linked back together to become ATP, goes somewhere in the cell to do some work, and continue this over and over again. So this enzyme called ATP synthase, sometimes people call it chemoosmosis because a chemical is going from one end to the other, the hydrogen ion, and through that process we're going to make a bunch of electricity, or we're going to make a bunch of ATP. But we have a problem, and the problem is that if these hydrogen ions aren't taken away, we're going to be in equilibrium on both sides. Hydrogen ions will be here, hydrogen ions will be here, and then this ATP synthase will stop. So we have to do something, and what we're going to do is we're going to have to make sure that we get rid of those hydrogen ions. And how we do that is we're going to do another pathway. Remember how we had those electrons and they were coming this way? And we made some from NADHs, and we made some from the FADHs. Well, what happens is when two hydrogens come together with an oxygen, this system is going to turn that into water, H2O. And water becomes the waste product that actually leaves this. And if we didn't do that, what would happen is that the hydrogen ions would be at equilibrium on both sides. Eventually this will stop. And when this stops, we wouldn't be able to make energy anymore. So this is crucial. And if you ever go for like a long hike or a run or whatever and start to feel these burns, they're called lactic acid burns. What's happening is you're not moving oxygen around your body very effectively. And what's happening is these cells are starting to stop these processes. And acid is starting to build up. And that acid is starting to cause a problem in your cells, and if not treated right away, will cause your cells to die. And basically what happens is the cell hopes oxygen comes. Usually that pain gets you to slow down, and that slowing down or stopping is going to make it so oxygen can move around your body more effectively. And then eventually when oxygen gets to these areas, everything gets healed. And then that acid is just chucked into the bloodstream as a waste product, and your liver takes care of it. So hopefully that makes sense. If I go through the PowerPoints, you'll see that the first thing, you can go through yourself and check it out. The electrons leave the coenzymes, those NADHs and FADHs. We're going to go ahead and have these special proteins that are going to move these hydrogens. We're moving it against the gradient, which is another way of saying high to low. So we've created a situation where there's more hydrogens on one side than the other. We have this high protein gradient on one side. Eventually what happens is they're going to go to the ATP synthase and come out. And that's going to make us ATP. Also, remember, we have to get rid of those hydrogen ions, so we add the water. And the water, we add the oxygen, and the oxygen binds to the two hydrogens and makes water, and that becomes the final waste product. In our next video, we'll talk about poisons.